Hello friends, welcome to Gluconeogenesis part 1, this module in which we discuss the substrates which are gluconeogenic or substrates which give rise to glucose through gluconeogenesis. Now, gluconeogenesis meets the glucose demand of the body when dietary carbohydrates are unavailable. So that is the main significance of gluconeogenesis. It happens or occurs mainly in liver cells, and we can go through the substrates one by one. Now, the normal level of blood glucose, blood sugar in our body is 70 to 110 milligrams per deciliter. And the importance of maintaining normal level of blood glucose is especially because our brain requires around 120 grams of glucose per day. 120 grams per day. This is out of 160 grams. Out of 160 grams per day of body requirement, because the rest of the body needs 40 grams per day. The glycogen reserve, we know that glucose is stored as glycogen for immediate release to glucose back to glucose again. The glycogen reserve can provide around 190 grams, and the rest of the reserves, like the glucose present in plasma or tissue fluids, will be 20 grams, which make up a total of 210 grams, which is available in our body. But what happens is that during long-term fasting or during strenuous exercise, this reserve, the reserve to 10 grams, will be depleted rapidly. So at such times, body turns to other molecules, especially non-carbohydrate substances, for producing glucose. So this is substrates for gluconeogenesis. We can discuss one by one. The first one is lactic acid or lactate. Lactate is produced in skeletal muscle cells as a result of muscle action as well as in RBC. This forms one of the most important of the substrates for gluconeogenesis, and this is formed from pyruvate. And as we know, pyruvate is formed through glycolysis from one molecule of glucose. It gives two molecules of pyruvate, and pyruvate is considered as the end product of glycolysis. And the pyruvate, the usual process is when oxygen is available, the pyruvic acid formed during glycolysis goes on to complete aerobic oxidation after getting converted to acetyl coenzyme A and then passing through TCA cycle, so that plenty of ATP can be produced. Plenty of ATP or energy can be available. But when oxygen supply is insufficient, typically during intense muscular activity, ATP must be released through anaerobic metabolism. And so this is the route that is taken in the muscle cells during strenuous exercises. Pyruvate formed from glycolysis will be converted to lactate by the action of lactate dehydrogenase. So what muscle cell actually does here is that this process can regenerate NAD+, which actually is a substrate for glycolysis. And during glycolysis, NAD+ gets reduced to NADH, and NADH, as we know, will be used up later. Here, if NAD+ is not readily available, glycolysis cannot proceed further, so that the net production of 2 ATP molecules from glycolysis will not be available for strenuous muscular activity. So that's why pyruvate gets converted to lactate, so that NAD+ can be regenerated and can be rechanneled to glycolysis, so that further ATP production can be carried out. So this process, which supplies enough of NAD+ for a continued glycolysis in skeletal muscles, but this leads to accumulation of lactate also in muscle cells. So this actually leads to what is called lactic acidosis and low pH. In fact, this enzyme LDH in muscle, it cannot reverse lactate to pyruvate because it is not thermodynamically favorable. So this is when CORI'S cycle come into play. Now, CORI'S cycle, as we know, glycolysis, anaerobic glycolysis, accumulation of lactic acidosis in muscle cell, and lead to muscle cramps and increase in blood lactate. Here, the lactate produced or accumulated in muscle cells is carried through blood to liver cells, because lactic acid can cross the membrane of muscle cells. It can pass through blood circulation and can cross the hepatocytes membrane also, so as to reach liver cells. So this is what happens. Lactic acid accumulates and in Cori cycle, this lactate goes through blood, reaches liver, where it gets converted to pyruvate. And pyruvate is, as we know, or as we will discuss later, it's a prime substrate for gluconeogenesis. So pyruvate, then gluconeogenesis produces glucose. So this process actually helps in efficient utilization of lactate. Besides production of NAD plus for glycolysis, the lactate gets converted to pyruvate and then goes through gluconeogenesis, because in an actively contracting muscle, only 8% of the glucose goes through aerobic glycolysis, and the rest 92% of glucose is used for anaerobic glycolysis for meeting ATP needs. So that's why the muscle cell needs continuous production of glucose through gluconeogenesis. And this is what happens. And Cori cycle. Carl Cori and Gerty Cori, husband and wife, jointly they won 1947 Nobel Prize in Physiology and Medicine. But it was not for the discovery of Cori cycle. It was for the discovery of the course of catalytic conversion of glycogen. Glucogenic amino acids are another source of glucose. Glucogenic amino acids can be converted directly to pyruvate or other glucose precursors. All amino acids except leucine and lysine here are glucogenic. Actually, leucine and lysine are ketogenic in humans, and amino acids are actually used during prolonged starvation and diabetes mellitus. When, especially during starvation, when the body first starts to use glycogen reserve, when that is depleted, turns to fat, and still prolonged starvation lead to utilization of or breakdown of protein, and amino acids will be used for gluconeogenesis. Now, this is an exclusive figure showing the conversion of amino acids at different stages of a TCA cycle, and of them, this one. These amino acids actually give rise to acetylcoenzyme and acetylcoenzyme. Even though acetylcoenzyme passes through TCA cycle, the carbon skeleton will be released as carbon dioxide at different stages here and here. So acetylcoenzyme is not considered as a precursor of oxaloacetate or as a precursor of glucose during gluconeogenesis. The rest of them, primarily this one, they directly give rise to pyruvate, as well as here alpha-ketoglutarate, succinylcoenzyme, fumarate, and oxaloacetate. All these are metabolic junctions where amino acids will contribute to formation of glucogenic substrates. The production of glucose from glucogenic amino acids involves these amino acids being converted to alpha keto acids and then to glucose, with both processes occurring in liver. Now, one of the most important glucogenic amino acids released from muscle is alanine. You can see glucose-alanine cycle here. Alanine released from muscle is a major substrate for gluconeogenesis, especially during starvation, and this is what happens in muscle cells. We know that pyruvate can get converted to lactate, which we have already seen. It can also give rise to alanine through a process called transamination. And the alanine, which is present in muscle, can cross the membrane of muscle cells and reach liver cells or hepatocytes, where the alanine will be converted to pyruvate with loss of an amino group. And this amino group, which will be converted to urea and will later be excreted out of the body. This process of conversion of alanine to pyruvate is called deamination. Let us see what are transamination and deamination are. Now, this is transamination. Transamination is the process by which amino groups are removed from amino acids and transferred to acceptor keto acids. The keto acid here is alpha ketoglutarate, to generate the amino acid version of a keto acid and keto version of a non-original amino acid. We will discuss transamination in a different lecture in detail. So this particular process is catalyzed by alanine transaminase here. And what is deamination? This is what deamination means. The amino group is removed from an amino group, from an amino acid. Deamination is the removal of an amino group from a molecule, like amino acid. Enzyme that catalyzes this reaction are called deaminases. Here, both the processes come into play during alanine glucose cycle. Now, fatty acids with the odd number of carbon is also a good substrate for gluconeogenesis. Oxidation of odd chain fatty acids actually produces propionyl consomme, a 3-carbon compound. Propionyl consomme is a good precursor for gluconeogenesis because it yields oxaloacetic acid. And propionyl consomme originates from catabolism of some essential amino acids also, like valine, threonine, isoleucine, and methionine, besides odd chain fatty acids. The side chain of cholesterol also gives rise to propionyl consomme. Now, this is a major pathway of conversion of propionyl consomme into succinyl consomme, as shown here, and succinyl consomme. And so, it is through succinyl consomme that this propionyl consomme enters TCA cycle so that it can get converted to oxaloacetic acid and go through gluconeogenesis. The biotin dependent enzyme propionyl consomme carboxylase, it converts propionyl consomme to d-methylmalonyl consomme with addition of a carbon dioxide, so that this product actually becomes a 4-carbon compound. As we know, succinyl consomme is also a 4-carbon compound. And d-methylmalonyl consomme, it is racemized into L-methylmalonyl consomme, which gets isomerized to succinyl consomme, the Krebs cycle intermediate. And even numbered fatty acids are not converted to glucose because they during catabolism of even numbered fatty acids, the product is acetyl consomme, which is not considered as a gluconeogenic substrate or as a glucose precursor. Glycerol is another very good substrate for gluconeogenesis. It is produced by hydrolysis or breakdown of a triacyl glycerol, and glycerol get into gluconeogenic pathway through dihydroxyacetone phosphate, DHAP here, and it can get converted to glyceraldehyde 3-phosphate and back to fructose 1,6 bisphosphate or biphosphate through aldol condensation, so as to give rise to glucose again. Now, glycerol actually undergoes, this is what happens, glycerol kinase gives L-glycerol 3-phosphate, which give rise to dihydroxyacetone phosphate. Glycerol phosphate dehydrogenase involved in the process, this enzyme. So this is how glycerol gets converted to dihydroxyacetone phosphate, and through DHAP it goes back to glucose. Thank you.